Hi, welcome to UCSF's first ever Facebook Live event. Today, we're here to talk with one of our scientists in our research labs here at UCSF to show how we're advancing health. I'm Kishore Hari. I work at the Science and Health Education Partnership here at UCSF, and I'm thrilled to be joined today by Professor Shubho Roy. He's a professor in the Department of Bioengineering and Therapeutic Sciences in the School of Pharmacy and School of Medicine, and we're here today to talk about the Artificial Kidney Project. Shubha, welcome to Facebook Live.
Kishore, thank you so much.
All right, so when we say Artificial Kidney Project, what are we talking about here?
Well, here is a prototype model of what we're going to talk about today. It's a plastic non-working prototype, but shows you the components of the artificial kidney.
Before we get into the technical details of the artificial kidney, which we're going to do, let's talk a little bit about the background and the need for this project. Why artificial kidney? What is sort of the basis of the medical need for a project like this?
Sure. So when kidneys begin to fail, they go through a series of stages. Many of you may not know this, but kidneys actually do a bunch of functions than just make urine. They have 7 or 8 different functions. And as you go through chronic kidney disease, you start losing kidney function. And when you get to the very end of that cycle, you end up at end-stage renal disease, or ESRD. At this stage, you basically have 2 options. Either get a kidney transplant or get on dialysis.
Now, dialysis is obviously a very difficult process. It's both emotionally and physically taxing to the patients and the patient communities, but it's also really expensive. So how does this idea of an artificial kidney resolve some of those issues?
Sure. So let's just take a step back and think about the problem. Today there are over 600,000 people in this country that have kidney failure. Worldwide, that's about 2.5 million people. This number continues to grow at about 5%, driven primarily by the epidemic of diabetes and blood pressure, hypertension. So most of these patients really need to figure out how to get a transplant. As we know, there's just not enough organs available. The vast majority are limited to dialysis, which is inconvenient, expensive, and ultimately not a very therapeutic process, meaning that it does not provide all the functions of a kidney. What happens is these dialysis patients end up not only experiencing poor health, but they also cost, in this country, a tremendous amount of money. Each dialysis patient costs Medicare, the insurance company that pays for treatment, almost $90,000 annually. In contrast, if one is lucky to get a kidney transplant, that number is only about $30,000 after the surgery. So today we spend about $35 billion on treatment of kidney failure. Interestingly, $35 billion is larger than all of the NIH budget that we spend on medical research.
So this is a massive economic issue in addition to just the quality of life and better outcomes for the patient that'll come. And I'm just going to jump in and say, if you have questions for Shubho, please post them in the comments below, and we'll have a Q&A later in this Facebook Live. All right, let's talk about the actual prototype itself before we get to a really special demo of it. How does the prototype work? It's kind of much smaller than I expected when I first heard the idea of an artificial kidney.
Sure. So many of us have heard of the term artificial kidney in the context of in-center dialysis, which is a big machine the size of a refrigerator. Now, we know much of the inconvenience and complications of dialysis is the fact they have to go 3 times a week, and they have to be connected through a catheter to a system that removes blood and purifies it. That has a number of challenges. So what we've decided is actually build an implanted device to provide treatment 24/7. Now, what do we have in this? We try to mimic more than just dialysis. We try to provide the function of filtration that dialysis does partially, but couple that with some of the cellular functions and biological functions provided by the proximal tubule of the kidney, and that's done through a cell bioreactor unit. This 2-stage system works in tandem to mimic more physiological performance of the kidney.
So, kidney, obviously, a very complicated organ. It has lots of functions, but you're really trying to mimic that filtration behavior, that purification of blood and other fluids, and then also the cellular function. Can you talk just a little bit more about what the function of those cells are?
Sure. So, in our kidneys, the proximal tubule performs a number of functions, including production of vitamin D, helping regulate hormone secretion and remove toxins. It also helps with the maintenance of pH of blood as well as immune protection. So by bringing in kidney proximal tubule cells into the bioreactor, will recapitulate those functions that just the mechanical filtration will not.
So let's take a look at the actual prototype that you had up before. Is it this red device here, or,
Yes.
or this?
So here is actually our early version, and I'll show this because it shows this clearly. This is the filtration component that just has a series of filters that remove processed blood and remove the toxins and water. The second part here has the cellular component.
And so let's break down some of these components now. So the filtration component, when we say filter, I think people think of like their home base, like, refrigerator filter. That's not how this works.
So this is actually it's slightly different. So let me show you what we have in here. We actually use silicon semiconductor technology, the same technology used to make electronics. So here's a silicon filter. So this is a flat sheet of silicon that we have patterned with very fine pores. These membranes are cut into sections, as shown here, and these sections are then assembled into packages of titanium that are here, and what these packages are then assembled into our little plastic prototype.
So they're in kind of columns of wafers,
Yes.
side by side?
Yeah, they're a parallel plate filter. Don't think of them as the home water filter used for purifying water. Blood flows over them, and then you generate ultrafiltrate as
So as the
it.
fluid is passing over the filter, some of it comes through, and that's the way it gets purified. I'm curious, like most filters that we're used to seeing at home have to have a pump system. Do you have to have a pump system here?
That's the beauty of using semiconductor technology. We can actually arrange these membranes so that they are parallel, which means that they have very low resistance, but also the efficiency of the silicon membranes is such that you can require very little driving pressure. The blood pressure between the artery and the vein is enough to generate the filtrate.
Oh, that's a huge advancement, that idea of using the body's own blood pressure to achieve some filtration effect. That's something that wasn't available even like a decade ago.
That is correct. So what makes this possible is by designing a membrane that's used as silicon, we can design it to have a very high permeability and a very high selectivity. These two work together to achieve a performance that's not possible with any other membrane technology today.
And for those of you who just may be joining us, we're talking with Dr. Shubho Roy about the Artificial Kidney Project here at UCSF. If you have any questions or comments, please type them below, and we'll be having a Q&A session in a little bit. Let's talk about the cellular component before we get to the demo, which I'm really excited about. So the cell component, what is that doing once it gets past the filter?
Sure. So what we have is we have the filter, the hemofilter that's generating ultrafiltrate. This ultrafiltrate consists of toxins, urea, beta-2 microglobulin, bad stuff that you have drank, for example. And then you've got this ultrafiltrate being sent over the cells. The cells are like Santa Claus. They know what's good and what's bad. And they're able to pick out the water, the salts, and the sugars and transport them back into blood, concentrating the remaining into urine that's directed downstream towards the bladder.
And when you say this whole device here, if you don't mind me holding it up, this whole device is this about the size we're talking about the finished product will be?
Well, Kishore, this is a good point you make. This was our initial sizings based on the technology about 3-4 years ago. But with improvements in silicon fabrication technology, we can actually make this smaller. So today, we have something that we estimate will be this size. But the current tests we are doing in the lab is through a filter just this size. So we hope that we'll get to something even smaller than what I just showed you, but we know from talking with our colleagues in surgery, this is actually possible to implant.
So the idea is to get down to the size of an actual kidney in a lot of ways. Like, so this implantable piece will roughly occupy the same space. Is that right?
Yeah, the idea is to be able to provide a surgically implantable device in the same location as a transplant is located. So you go in, a surgeon performs the surgery to surgically connect the device to the artery and the vein, close you up, and then you're able to get the continuous treatment provided by the artificial kidney.
So today, Shuvo actually set up a demo of how this functions, so you can see how the blood gets translated through this filter, through the cellular matrix on the other side to be purified. So let's
That's
take a look.
correct. So thank you for giving me the opportunity. So let's just show you what's here. So what we have here is a mock circuit, and I'll move this out of the way. This is supposed to be our dirty blood.
And what is in there, just for reference? It's not blood.
That's
today.
right. It's just some coloring, food coloring. And what we have here is a pump. It's a lab pump, but it mimics the heart.
So this is about at the same pressure our heart would be pumping blood at.
That's right. Okay, and then we are mimicking this pump, passing it through an artificial kidney, which
So
is
it
shown
goes
here.
So the first stage it goes through is the filter.
That's correct.
And then as it passes through the filter, it goes through that cellular matrix. Is that the right word?
That's correct. It goes through the bioreactor, of
Bioreactor.
cells, and it sends it back into the bloodstream.
Mhm.
So just like our own kidneys, we've got blood coming from the heart, going through the kidney, getting filtered, and going back into the heart. But we also have a third port here that's generating the urine.
Mhm.
And you'll notice that this is yellow, and we are cleaning up this blood. And if I give this sufficient time, this would actually become all clear. And all the yellow fluid would be packed up here.
So this is basically, in its current iteration, running at essentially the same speed our kidney would be running at. Is that right?
Yeah, this is actually, for our purpose of demo, I'm actually running it slower. But in practice, this is almost the same, but we are going to speed it up.
Got it. And I'm used to having to replace filters. Like in my kitchen, in my home, filters have a lifespan. Does this device, would it also have a lifespan, particularly on the filter side?
So obviously that's a great question, and we are hoping that we have come up with strategies to minimize the need for replacement of filters. So we've coated these membranes with thin polymer coatings that prevent proteins and other blood components from depositing in the filter and gunking it up or causing a blood clot. However, as engineers, we have to think and be prepared for what may be a catastrophic problem, which is what happens if that does occur. What we'll do is to make sure that we will have a strategy to replace the filters through a minimally invasive procedure.
So the idea is that the surgeon will be able to go back in, replace this, but not in a way that is going to require extensive hospitalization or an extensive amount of time and money, as would be the case with a dialysis situation right now.
Yeah. What we'd hope is that you'd have one major surgery to implant the filter, and subsequently you would actually go for a minor surgery to replace the filters if need be, or the cells.
Yeah, so the same applies to the bioreactor then too. It also has a lifespan, but the idea is that it would be able to be replaced and replenished as needed.
The cells would have to be replaced or replenished. Hopefully we don't have to dismantle the whole device, and that's part of our engineering strategy.
Now, when it comes to talking about organ transplants, one of the words I always hear is making sure the donor can accept it, that the donor is not going to reject it, that they have to be on a regimen of immunosuppressants for a long period of time. How does this deal with that issue? Because the idea of an artificial organ being implantable in anyone is a huge hurdle to overcome.
So when you implant cells or transplant cells, you really have to worry about organ rejection. So we try to overcome this using the membrane itself, the membrane on which the cells are being grown, allows for nutrients and waste to go through the membrane, but the pores are too small for the body's immune system to attack these transplanted cells. So this strategy allows us to offer a treatment in where no immune suppressant drugs will be needed.
Let's talk a little bit about how you got interested in this. Because I kind of expect that you didn't start out graduate school saying, I'm going to work on artificial organ development. Can you tell us a little bit how you came to this, this problem and this solution?
Sure. So yes, you're absolutely right. My background is in physics and engineering, and I wasn't planning to necessarily work on the kidney. But what I did was to meet up with clinicians that told me, can you help improve a dialysis machine with your engineering background? As we started looking at this problem, one of my colleagues, William Fizel, who's at Vanderbilt University now, identified that the membrane was a key challenge in improving dialysis and making it more portable. So he and I established a partnership that then said, if we can make a better dialysis machine, that's not enough, but we'd actually have to provide the other functions of a kidney. So over the years, I've brought my engineering skills, and he's provided his clinical skills to the point we are today.
I love that aspect, especially you see that a lot here at UCSF, where you see multidisciplinary approaches to problems. It's not just biologists and physicians tackling problems. They're bringing in engineers and physics to deal with some of the novel issues of the day. We're going to take questions here in just 1 minute. I had one more question before we go to the Q&A for you, and that's I want to talk just briefly about the technical leaps that have been made in order to get to this prototype stage. Because one of the big issues that we see with anytime you're applying filtration technology into the human body is the miniaturization of it itself. I mean, a lot of what we've seen, the reason it was done via dialysis is we needed that big space to handle it. What has been, you showed us the silicon wafers before. Is that the big advance that has allowed us to miniaturize this component in a way that allows it to be implantable now?
Absolutely. So I think the big innovation on the miniaturization side is to make the membranes much more efficient. So a typical dialyzer has about 1.5 to 2 square meters of surface area. We can achieve that same amount of filtration, or clearance if you will, in about 1/10 or 1/20 of that surface area. And that allows us to build something that's a lot more compact, which allows us to make it implantable.
We're going to start taking questions now. The first question comes from Khan, and his question is that the final shape that we're going to see when it comes to the implantable kidney?
What a great question. And the truth is, when I started the project, we, with our engineering team, started with something that kind of looks like a car battery, as you can see. But we started getting feedback and from some of the patients and patient families, and we're now at a shape that's a little more rounded, a little more organic, but by no means final. A lot of this design is based on our engineering capabilities, but also patient preferences. So what we hope is over time, it'll look a little more organic, a little more rounded, and we're actually going to get input through our tests of surgical technique that will help us define the final shape.
I have to say, I like the logo on it, though. So if you can keep that in place. Before we take our next question, I was wondering, what stage of the project are we at now?
Yeah, sure. So we have done a lot of the benchtop testing, as you've seen here. We've designed it. We're now in the process of scale-up. We're doing preclinical testing, evaluating the performance of the filter in vivo. We're testing the longevity of the cells on the benchtop and then moving it in vivo. Once we have established that, we'll bring the two components, the hemofilter scaled up with the bioreactor together and demonstrate its functionality and safety in a preclinical setting before we move on.
We have another question from Sylvia in Colorado, and she asked, if I live in Colorado, can her doctor refer her to the kidney project when it's ready? Is it going to be in that situation where we're going to get referrals from all across the country?
Yeah, another good question. At this stage, obviously much of the development is happening in a couple of areas around the country, but our plan is when this device is done, it should be available everywhere and wherever it's needed. That means that somebody in Colorado could get it, or logistically they could come here and get it. But obviously we're a little far ways from that right now.
Yeah, let's talk about the pace and history of this before we take a few more questions. And we're going to spend some extended time today because there are a lot of questions coming up. The idea of artificial organs has really become very much in the science news over the past couple years. But I want to temper that expectation that it's here tomorrow with the pace that it's actually going at. Can you give us an idea of how long this project, when this project started to where we're at now, to some beginning projections of when we're going to actually see it progress to those clinical trials?
Sure, sure. I mean, so obviously I've been working on this project for an extended time now. The initial discussions with my colleagues started in the early 2000s. At that time, we conducted some early exploratory research. And it was not until the late 2000s we felt we were onto something. And in 2007, with the support of NIH, we were able to take the first major step in making a prototype. Now, since then, we've had to go through a series of steps that let us answer both scientific and engineering questions. But ultimately, what we'd like to do is to provide a treatment that's safe and has efficacy. We do not want to compromise that. That does take time. We have to establish the device is not going to clot, the cells are going to live. So what we see here does not readily translate into a clinical trial immediately, but we have to go through a series of carefully calibrated steps, and once we do that, we get to the clinical trial. To give you a sense for our timeline, we hope to finish the preclinical testing sometime towards the end of next year.
Oh, that's very soon.
Yeah. Of course, assuming everything goes well, and we are keeping our fingers crossed, then we'll start the initial clinical trial for safety that shows that no blood clots, the device is safe, and if everything goes according to projections, we anticipate it will be towards the end of this decade that will be available to the public.
Well, and so when you look at that in whole, we're talking about a little more than a decade from the first ideas of this to it actually being hopefully a reality. Knock on lab bench top. Is that what you knock on for luck? Is the lab bench top?
Yeah.
We're going to take a few more questions. You're open to that?
Of course.
So Danny asks, how much global peer review is involved? And I guess there's a larger question there too. Is there must be a number of collaborators beyond yourself in this project?
Sure. So we have a team that is based at UCSF with a key group at Vanderbilt, but of colleagues at the University of Michigan, in Ohio, in Pennsylvania, most of them still in the United States. We are starting collaborations across the world. In terms of the amount of scientific input we get them through a series of avenues, including peer-reviewed journals, but also when we apply for our grants from NIH, they have to go through a rigorous review process. So that allows us to also tease out the key questions that have to be answered and before we can get our funding to move forward.
Nikki asks a really great question. Is this device better than a transplant itself?
Another great question that I really think deserves a thoughtful answer. So a transplant is a natural kidney. It provides a host of functions. If you recall, I said we have about 8 to 10 different functions a kidney provides. Dialysis provides one just and by itself and not that well. We'll provide probably some of those functions, not everything like a transplant, but well more than dialysis. And I think if we do that, we'll allow patients to live a more healthy life that's also better for them and their family members.
That's great. Margaret asks, is this going to be available internationally, or is this going to be a U.S.-centric development?
Margaret, we're developing it right now here, but our plan is to make it available anywhere and everywhere it's needed. Obviously, that will require partners. It will require different logistics to make that happen, but we have no intention of just keeping it for the U.S.
For those that really want to learn a lot more about this project, I encourage you to visit kidney.ucsf.edu. That has an entire history about the project, some incredible animations that Shuvo and his team has put together, and an articulation of some of the timeline, the next steps. And speaking of next steps, we have some questions about how someone can participate in the forthcoming clinical trial.
Sure. So we have a number of ways people can get involved. One is just to raise the awareness of what we are doing. Part of what is important is to let the patient community and their families know that this is something that's being worked on, and we rely on people's support to advance our research. The way to get involved is go through our web page, kidney.ucsf.edu, sign up through our Facebook page on a clinical trials waiting list. So when we are ready, you'll be aware, and you can then take the next steps.
And that Facebook group, for people that want to know, is called facebook.com/artificialkidney. I encourage you to go there. There's a whole community of individuals that are interested and engaged around this topic. What are the next steps to make this a reality?
Well, the next steps for us is really to complete the preclinical testing. And that's dependent on a couple of things. One is the results, obviously, that come up in the bench testing, and we are very fortunate that that looks very promising. The other one is the amount of resources we can bear. Some of that is financial, some of that is just collaborations. So by bringing people who are motivated to work on the project, we'll move to the next step in terms of scaling up the filtration component, making sure the cells live longer, build prototypes of increasing sophistication, test them, refine the design, test them again, and then get to a point where we have sufficient data to take to the Food and Drug Administration to get approval for the first clinical trial.
Now, while this development of the artificial kidney is mind-blowing, it can make a huge difference to so many people's lives, I'm wondering if you zoom out even further, are we going to see these types of projects for other artificial organs as well? This sort of cross-collaboration leading to novel engineering and designs of other types of artificial organs. Is what you're doing here going to affect other fields as well?
Well, I can tell you some of the experience we've had here at UCSF. As we pulled a team of engineers, scientists, nephrologists, surgeons all together, we find that the technology we are implementing for the artificial kidney may be adapted for other applications. So one spin-off project from the lab has been diabetes. So we've had some pancreas transplant specialists who have come to us and said, if you're able to put insulin-producing cells in your device, maybe you could cure type 1 diabetes. So there's a small group at UCSF that's exploring that possibility now.
That's the best way. That's the best news of all, because to see like this have tendrils that go out into other areas of scientific research, that's why we do all of this, right? To really advance the knowledge and really help patients down the line. Thank you so much, Dr. Shubha Roy, for joining us on this first ever UCSF Facebook Live event. We hope to take you into more labs and meet more of our scientists that are innovating and changing the shape of healthcare in the future. So stay tuned to the UCSF Facebook page for more information on that. For more information on the Artificial Kidney Project, visit kidney.ucsf.edu or to their Facebook page at facebook.com/artificialkidney. But please join me in thanking Dr. Shubha Roy again for taking us on this amazing tour of the Artificial Kidney Project.
Thank you. Thank you.